I will be discussing first the disorders associated with decreased protein energy consumption. So this condition is actually considered the most widespread nutritional problem common in undeveloped and developing countries. And this basically affects children with prevalence varying from 20 to 50% in different areas. And since this is rampant in developing and undeveloped countries, basically the major risk factors are as follows. So we have a very low socioeconomic status and low level of education. So malnutrition is actually a deterioration of nutritional status over a short period of time, and this is usually characterized by wasting or nutritional edema. So wasting is represented by acute weight loss or reduction in muscle mass, whereas nutritional edema is characterized by presence of bilateral pitting edema, initially involving the legs and both feet, which can progress to become generalized all over the body. As to classification, we have this what we call moderate, severe, and global acute malnutrition. And severe malnutrition actually have three clinical forms. This involves is what we call marasmus, which is associated with severe weight loss or severe wasting. We also have this what we call kwashiorkor, which is associated or with nutritional edema. And we have a combination of both marasmus, kwashiorkor, which is a combination of severe wasting and nutritional edema. Okay, so let's start off with marasmus. This is a Greek word which means to waste, and this basically results from severe deficiency of both dietary energy and proteins, and this is characterized, as I've mentioned, by muscle wasting. So what happens in marasmus is that as the body utilizes stored substrate or substances to produce energy during periods of nutrient inadequacy or starvation, it basically results in severe loss of subcutaneous fat and muscle. Okay? So again, let me just explain it in a simpler terms. No? So what happens in marasmus is that your body actually tends to find alternative sources of energy. And in the process of getting other sources of energy, especially during starvation, when you have inadequate food or protein intake, so basically it causes loss of your fat and muscle. So it basically consumes your fat and muscles to produce energy. Okay, so this is now called, that's why it's called wasting, or marasmus, or to waste. And this is commonly found in nutritional emergencies, conflicts, and during calamities, or times wherein there is prolonged starvation. So the primary cause of marasmus is a type 2 nutrient deficiency in very poor quality diets that results in loss of appetite, growth cessation, and lowered immunity. And as a consequence, your body, as it conserves energy, it reduces physical activity, appetite, and consequently growth retardation. Okay, and another condition associated with severe malnutrition is kwashiorkor. So this means sickness of first child, and this is common, common in places in Africa, and this is actually characterized by edema, in contrast to your marasmus, which is characterized by severe muscle wasting. So kwashiorkor, beyond being caused by protein deficiency is actually precipitated by environmental insults or noxae, which basically increases free radical formation. And these free radicals containing a single unpaired electrons are highly reactive and frequently results in toxic effects. And environmental causes of your kwashiorkor may include those conditions associated with infections, exogenous toxins, contaminated foods, or small bowel overgrowth. So this is a picture, no, showing to us patients with marasmus on the left and kwashiorkor on the right. So notice the characteristic wasting, muscle wasting in marasmus, whereas in patients with kwashiorkor actually characterized by bilateral, no, or generalized edema, no, all over the body. Several factors actually causes your protein energy malnutrition. And this includes improper, no, the most common one, improper or inadequate food intake. Also, diseases causing inadequate absorption of food, metabolic abnormalities, emotional factors, and deficiency of food supply or poor dietary habit. So in evaluating for protein energy malnutrition, we look at, no, the weight of the patient or age. And usually these patients with protein energy malnutrition or PEM are underweight. And if we compare the height of the patient for age, usually this is associated with stunting, or stunted growth. And if we plot out weight for height growth of the patient, this is usually associated with wasting. We also check for the head circumference of the patient, and we measure the mid-arm circumference, no, and the skin fold thickness of the patient. So this table is lifted from WHO, no, classification of malnutrition. So kwashiorkor is associated with 80 to 60 to 80% of body weight compared to standard weight, with associated edema and deficiency in weight for height. Whereas your marasmic kwashiorkor, or a combination of both, is associated with edema and deficiency in weight for height also. However, the percent of body weight compared to that of standard weight is less than 60%. No? And in marasmus, there's no presence of edema. Nutritional dwarfism is associated with less than 60% of decrease, no, body weight compared to the standard weight, with no associated edema and deficiency in height for weight. Whereas for underweight child, this is just associated with 60 to 80% no, decrease in percent body weight as compared to a normal or a standard weight. So in patients who are wasted, no, with muscle wasting, or patients with marasmus, since this is associated with loss of your subcutaneous tissue, your loss of muscles, so you actually get to see prominent bones, no? As in this patient, the ribs are prominent. And as I have mentioned, the patients with kwashiorkor actually presents first with bilateral edema. And this edema in the feet basically progresses to edema in the hands, in the other parts of the body. Okay, so let's discuss briefly the biochemical mechanisms associated with marasmus and kwashiorkor. So again, as I have mentioned, what happens in marasmus is that there is inadequate calorie intake or deficiency of intake. Basically, may be caused by different factors, no? Maybe poor dietary intake due to a certain disorder, or there's really a low supply, no, dietary supply, or poor dietary habits of the patient. What happens is that for the body to compensate, or for the body to actually get adequate calorie, is that this causes decrease in insulin levels and increase in cortisol levels in the plasma. And this decrease in insulin and increase in cortisol results in protein breakdown and release of amino acids from the muscles. What happens is that the protein stores in your body actually undergo proteolysis or breakdown. And eventually, what happens is that when these proteins now are broken down or when they undergo proteolysis, it is producing, the simplest form of molecules or substances which make up the whole molecule of proteins. So we call it amino acids, no, which are actually the building blocks of your proteins. And repeated repeated breakdown of your protein stores and continuous release of your amino acids from your muscles basically leads to severe muscle wasting. And if this continues to happen, your liver will become now depleted with the necessary amino acids which are used to synthesize for other types of proteins, for example, your albumin. Okay? And since, remember in your anatomy and physiology, you learn that your albumin is actually a major contributor to your oncotic pressure in the plasma. So what happens is that for your oncotic pressure to be maintained, your albumin actually causes, okay, what does it do to maintain oncotic pressure? Is that your albumin produces a negative gradient such that whatever fluids now or the plasma or the serum in your circulation now do not leak out. Okay, do not leak out or do not accumulate, okay, in your surrounding tissues. And since there is depletion or decrease or deficiency in your production of your albumin in marasmus, what happens is that it does not result in any forms of edema. Okay, so as you can see here, um, the picture on the left, no, is actually showing you how the mid upper arm circumference is measured, no, using this particular equipment. Okay, and whereas the patient on the right actually is skin and bones, no, so showing us a severe form of marasmus. Your kwashiorkor, on the other hand, okay, what happens there is that there is actually deficiency of protein or there is a decrease in the protein energy ratio. Okay, this means that you actually consume more calories than what you actually have in the first place. So your protein stores are actually overwhelmed by the number of calories consumed, no, in kwashiorkor. And so this leads to increased insulin levels and decrease in cortisol in the plasma, causing increased uptake of amino acids by the muscles, diverting them from the liver. And so since there's not enough amino acids now to cause synthesis of any other proteins, larger molecules of proteins, okay, this also reduces or causes a decrease in the synthesis of your albumin, leading to edema. And this also decreases synthesis of lipoproteins, as seen in fatty liver. And since in your kwashiorkor, there is increased production of your free radicals, this basically leads to formation of dermatosis or skin lesions. Okay, so look at the patient with kwashiorkor. So patients with kwashiorkor actually characterized by patients of presence of bilateral or generalized edema. Okay, that is the most distinguishing feature, no, between marasmus and kwashiorkor. The edema in kwashiorkor and wasting in marasmus. And notice also the presence of lesions is in skin lesions in these types of patient. So to compare between the two, so marasmus is common in patients below 1 year old, whereas kwashiorkor is common in patients 1 to 5 years of age. And when we say of marasmus, since in marasmus there's actually inadequate intake of your protein sources, no? So this actually results in a deficiency of calories. Whereas your kwashiorkor, the problem here is actually the decreased levels of your proteins in general. And your marasmus is also characterized by, no, actually both, no, are characterized by by growth retardation. However, growth retardation is more marked in your marasmus as compared to your kwashiorkor. And since these types of patient are actually calorie deficient, and so they appear irritable and frightful, no, or fretful. And since your kwashiorkor patients actually have more or less a decreased protein synthesis, no, so they are characterized by, or they appear clinically as lethargic and apathetic. Okay, and to add, your marasmus is characterized by shrunken, dehydrated or dry skin, and your patients with kwashiorkor looks plump due to the presence of edema. As to appetite, your patients with kwashiorkor are anorexic, and as other associated features include, for your marasmus, we have watery diarrhea and muscles are weak and atrophic, and for your kwashiorkor, we have angular stomatitis, chelosis, and also water diarrhea in the muscles. As to the level of serum albumin, remember, both are characterized by decrease in formation or production of your serum albumin. However, for patients with kwashiorkor, there's actually marked decrease in the production of your albumin, as characterized by less than 2 grams per DL of your serum albumin. And in marasmus, on the other hand, is associated with increased cortisol levels, whereas your kwashiorkor is associated with decreased cortisol levels. So other biochemical alterations in protein energy malnutrition are this what we call, okay, I've mentioned this already, hypoalbuminemia. Why hypoalbuminemia? Remember, in your PAM or protein energy malnutrition, there's actually decrease intake or inadequate intake of your protein or calorie, such that your calorie consumption now is overwhelms your protein stores, no, originally. And so this will actually lead to decreased amino acids now available for synthesis of your albumin, and so leading to hypoalbuminemia. And then this is also associated with decreased increase IgG levels, okay? So increased IgG levels due to associated infections. Remember, your antibodies, your different cytokines, or your different other, for example, your complement system or any other substances in your immune system are actually also comprised of proteins. And in your protein energy malnutrition, there's actually decreased levels of these types of proteins. And so what happens is that a decreased production of these immune proteins would actually predispose the patient to infections. Okay? That's why presence of infection would actually increase production of your IgG, immunoglobulins. Okay? And kwashiorkor, as I have mentioned, is associated with fatty liver, however not seen in marasmus. There's actually no associated changes in your tolerance to glucose, but hypoglycemia may be seen in marasmus. Patients are hypokalemic and dehydrated if there is associated diarrhea, and hypomagnesemia also is a common usual finding in patients with protein energy malnutrition. So usually we manage this by protein replacement, or calorie replacement of the patient. So the optimum response is observed with diets providing 150 to 200 kcal per kilogram body weight, and 3 to 4 gram of protein kilogram body weight. And we also give a dietary supplement of your proteins, no? So we may prepare it by mixing 3 parts of vegetable proteins and 1 part of milk protein. And usually these types of patients are monitored, no? Response to therapy are actually monitored by taking note of any disappearances of the symptoms initially presented by the patient, for example, edema. And we measure the ultimate response goal is to actually get an increase or normalization of the level of your serum albumin and a gain in the weight of the patient. And if and only if there is continued protein calorie malnutrition, if there's no treatment done, prompt treatment done to the patient, so this can lead to permanent or irreversible physical and functional deficits, okay, which actually have deleterious effects on the intellectual capacity in later life. And there may not be any sequelae where the moderate and mild forms are corrected in time. So for patients presenting with PEM, actually prompt or immediate management to the patients is very important. No, that is to avoid any permanent side effects or functional deficits of the patient. We have also what we call cachexia, secondary to any other disorders. So probably in patients with cancer, AIDS, or any other terminal disorders. This is actually just similar to marasmus. However, the loss of protein is more than that seen in simple malnutrition. And a common pathophysiology of patients presenting with cachexia starts with the infection of the patient or any other disease condition, long-term disease condition of the patient. That secondary condition predisposing to cachexia will actually induce production of inflammatory cytokines. These cytokines will eventually lead to breakdown of your proteins and then causing now increased energy expenditure. The basal metabolic rate or your BMR is considerably increased in patients with cachexia. And aside from causing proteolysis or breakdown of proteins, your cytokines actually also stimulate uncouplers. Okay, for example, your thermogenin. These uncouplers will actually cause oxidation or thermogenesis without trapping energy and eventually leading to cycling of your lipids. Okay, so free fatty acids are liberated from adipose tissue. So what happens in cachexia is that aside from proteolysis, there's also lipid lysis or lipid consumption. And these lipid consumption or breakdown of your lipids will also lead to liberation or production of your fatty acids that initially comprise your lipid stores. And basically, most of the tumors preferably use anaerobic glycolysis, no, ending to production of lactic acid. So normally, we use ATP as the energy currency, as a source of energy from our body. However, in cachexia, what happens is that instead of producing a lot of ATPs, less ATPs are produced because of the production of your lactic acid. Now, our second discussion would be all about the disorders now associated with the different amino acid metabolism. So we usually term these conditions as errors of metabolism, and they are considered inborn because mostly of these amino acid disorders are actually inborn. No, you're actually born with it. No, that's why it's termed as inborn errors of metabolism. And majority of these inborn errors of metabolism are actually inherited via an autosomal recessive pattern. So I will explain that in detail later. So this is actually a group of diseases that affect a wide variety of metabolic processes. Okay, so we actually have three targets. No, the inborn errors of metabolism actually have three affected targets. So it may be number one, there is defective processing of the amino acids or the protein. Or number two, there is defective transport of amino acids such that these amino acids now are not actually utilized by the body, could not be utilized by the body. Or there is a defect in the activity of an enzyme, which is actually its main function is to cause degradation or processing of that particular amino acids. And so any defect of these enzymes would basically cause errors now of metabolism of these amino acids. So the these are the predisposing factors which may cause to an impaired metabolism of your amino acids. So we have this what we call number one, deficiency or absence of a particular enzymatic activity. Okay? So remember, your proteins in the body undergo metabolism or the process we call proteolysis, eventually leading to production of this what we call amino acids. Okay. Now, your amino acids actually are utilized by the body for different processes. For example, a build-up, no, or formation of another form of proteins. And this formation of other proteins or any other processes will actually require catalysts or enzymes to actually cause, no, or to hasten these particular processes. And so any deficiency or absence of these enzymes would actually cause these amino acids not to be metabolized or processed by your body. Okay? Now, another pathophysiology involving your inborn errors of metabolism is a production of toxic metabolites as a result of build-up. And so remember, for example, in your enzymopathies or enzyme deficiency, what happens is that a particular amino acid is not actually processed. And so if these amino acids are not processed, what happens is that they build up, no? They increase in number because they are not utilized in the process of metabolism. Because in the first place, in inborn errors of metabolism, you have deficiency of enzymes which would metabolize that particular amino acid. And so that will eventually lead to build up, accumulation of these amino acids, eventually leading to production of toxic metabolic products. Another cause of your impaired metabolism is a secondary nutritional deficiency. Okay? So we actually need cofactors or any other substances that will actually aid or help the different enzymes to process or metabolize the different proteins and amino acids. And any deficiency of these substances, which will actually serve as your coenzyme or cofactors, will actually lead to impaired metabolism of your amino acids. Okay? Now, production of precursors accumulated due to block. Okay? So the block may be actually due to decreased production of your enzymes, or when there is impaired feedback inhibition, or when there are changes in the binding site or cofactor. So this is the outline for today's presentation. We start off by introducing to you the different amino acids found in the body, and I will discuss briefly how these amino acids are normally metabolized and what happens when there is any deficiency or any impairment in the metabolism of these amino acids, leading to a particular disease entity or any disorder. Okay, so this is a figure showing to us the different 20 common amino acids. This is actually 20, no, when the PowerPoint was made, but for update purposes, we actually have now 22 amino acids. And I will leave that as an assignment to you. Now, what is the 21st and what is the 22nd amino acid? That will surely come out in your examination. Okay? So as to classification, we have this what we call aromatic amino acids, branch chain amino acids, and polar amino acids. Under aromatic amino acids, we have phenylalanine, tyrosine, and tryptophan. Branch chain amino acids include valine, isoleucine, and leucine. And under polar amino acids, we have acidic and basic amino acids. So under acidic, we have aspartic and glutamic acid. Under basic, we have arginine, lysine, and histidine. So your body metabolizes amino acids in several ways, okay? So I want you to look at the diagram shown here. So normally, your triacyl glycerols during the process of metabolism are broken down into several components, namely glycerol and fatty acids. Let's start off with the fatty acids. Your fatty acids be, may be brought to the liver or may be brought to your muscles for further breakdown to produce energy. Okay? So your fatty acids has two fates. First one, it may be brought to the liver, and in the liver, it is broken down into carbon dioxide and ketone bodies. Or your fatty acids may be brought to the muscles wherein it is broken down to produce ketone bodies. Now, whatever ketone bodies produced from breakdown of fatty acids in your liver may be brought or transported by several chaperone proteins or transport proteins to the muscles or to the brain for conversion or for energy conversion. All right. Now, let's look at what will happen to the glycerol component of your triacyl glycerols, okay? Now, your triglycerol are commonly brought down or transported to the liver. And in the liver, your glycerol are synthesized, are broken down to produce a common energy source of our body, the simplest energy source that can be utilized by our cells called glucose. Now, the glucose is brought to the muscles as a source of energy. So basically, further metabolism of your glucose will produce ATP, and your ATP will actually serve as an energy currency, by your cells. Or the glucose may be brought to the brain or transported to the brain for further metabolism for energy production. Now, in the muscles, remember, we also have another macromolecule, which is what we call proteins. Now, these protein stores in the muscles, during metabolism, the proteins are actually broken down into its simpler building blocks, what we call now amino acids. Now, these amino acids undergo several processes. Eventually, I will discuss these several processes in the next few slides, but what I want you to think now about is that the proteins are broken down into its simplest building blocks, the amino acids, and eventually, via several processes, your amino acids become now or are converted into keto acids. Your keto acids are transported to the liver, and in the liver, the broken down form of protein, the amino acids and keto acids are converted to glucose such that the glucose now will be your sole provider of energy for the different parts of the cells. Okay? So this is how the amino acids are converted into keto acids, no? So again, generally, your proteins are first broken down into amino acids in the process of proteolysis. Now, the end product of protein breakdown produces amino acids, and eventually, when these proteins now are actually further broken down, it may produce several waste products we call now ammonia and urea. Now, these products of protein breakdown are actually recycled in the body for the generation of energy. So remember, your amino acids that I have mentioned in the previous slide are brought to your liver, and in the liver, they are actually converted into glucose, and that glucose now will be utilized by your cells as a source of energy. Now, some of the amino acids are further converted into alpha keto acids. And via several enzymes, okay, the individual amino acids are basically distributed to the different parts of the body where it is intended to be used. So why now your amino acids? How come now amino acids become alpha keto acids? It undergoes several processes. No, first it undergoes transamination. Trans means transfer. Amination means amine group or amino group. So what happens is that there is a transfer of amine group to another amino acid to form keto acids and another or new amino acids by this reaction is catalyzed by the enzyme transaminases or aminotransferases. And another process that it undergoes is a process of deamination. D means removal, amination or amine or amino, meaning you remove amino group from the amino acid or keto acids. Okay? And the enzymes which catalyze this reaction is called deaminases. This is followed by a process called transdeamination. Okay? So this actually involves a two-step process. First, there is transfer of amino group, followed by removal of the amino group, giving now an end product, ammonia, no? Ammonia, to give glutarate and free ammonia. Eventually, if metabolism of your protein continues, what happens is that these glutarate and ammonia are decarboxylated or they undergo a process called decarboxylation, meaning D, we remove carboxyl, we remove a carboxyl group from this glutarate and ammonia, eventually leading to production of CO, removal of CO2 no, to produce a particular amino acid amine. Now, this diagram will just actually show you, summarize everything that I've mentioned a while ago. No need to read this, no? This will not be included in the examination. Now, I mentioned a while ago that the different disorders of protein metabolism are actually inherited in an autosomal recessive pattern. What does that mean? Now, when you say of recessive, I'm actually talking about how these traits are passed. No, recessive would actually mean that the trait is actually not expressed by a particular person, meaning you have that trait, however, you do not develop that certain disorder for that trait, but you still carry that trait. That's why if this is inherited as an autosomal recessive, there must always be a carrier. So in the picture here is shown here is a picture of a paternal carrier and a maternal carrier, meaning the trait that they have inherited is just recessive, meaning not being expressed, no, meaning the father and the mother here actually do not have the disease condition, but they are actually carrier of the genes, which when become activated, no, when become expressed, cause a disease entity. So the check mark here would actually mean this is the normal functioning gene of that particular person, whereas the X mark here is actually the mutated gene. Okay? So when you do Punnett square or crisscross or mating or breeding, what happens is that this will actually eventually lead to 75% of the total number of offspring not having the disease condition. So the first child no, has inherited 2 working genes, whereas the second and the third child has inherited 1 working gene and a recessive mutated gene. So since it is recessive, meaning these offsprings will not actually produce the disease, will not have the disease, although they are a carrier of the non-functioning gene. Now, 25% or 1 of the 4 offsprings of these parents would actually would actually inherit both the mutated or non-working gene of the mother and the father. And since these offspring do not have any percentage of working gene, and so this offspring now will actually have that disease entity specific for that mutated gene. So we have 3 types of amino acid disorders. We have disorders of metabolism of aromatic amino acids, branch amino acids, and amino acid transport disorders. So let's start with the different disorders of aromatic amino acid metabolism. So most common conditions are most common aromatic amino acids with particular disease entity are phenylalanine and tyrosine. Okay? Any defect in the metabolism of your phenylalanine would lead to a condition called phenylketonuria. Okay? This is due to a deficiency or absence of the enzyme phenylalanine hydroxylase. Phenylalanine hydroxylase, which will convert phenylalanine to tyrosine. So I will explain that in detail later. Now, tyrosine metabolism defect would actually lead to several disorders. So we have alkaptonuria, albinism, so we have DRD and tyrosinemia. So I will explain this also one by one later. So let's start with phenylalanine metabolic disorder or metabolic defect. Okay? Now, normally, in amino acid or degradation of your phenylalanine, what happens is that your phenylalanine are actually converted into several metabolites. So in the first step, no, phenylalanine, due to the enzyme phenylalanine, is converted into tyrosine by the enzyme phenylalanine hydroxylase. Tyrosine, in turn, is converted by the enzyme tyrosinase into melanin. So your melanin now is responsible for the brown color or pigment formation, no? Responsible for the pigment formation in the skin or melanocytes. Now, your tyrosine, if not converted to melanin, may be converted into catecholamines and acetyl-CoA fumate. Now, the enzyme responsible for converting tyrosine to catecholamines, or catecholamines are actually epinephrine and norepinephrine, is this what we call tyrosine hydroxylase. Now, on the other hand, tyrosine may be converted to homogenesic acid and then to acetyl-CoA fumarate. Now, aside from being converted to tyrosine, your phenylalanine may be converted into phenylactate. So phenylalanine is converted to phenylpyruvate and phenylactate. So I don't want to confuse yourselves. What I want you to remember are the common enzymes which are defective in this metabolic process. Okay? Now, if the enzyme phenylalanine or hydroxylase is decreased in number, or if there is absence or deficiency of this enzyme now, what happens is that your phenylalanine is not converted to tyrosine. And so, if phenylalanine now being not converted to tyrosine due to a defect in phenylalanine hydroxylase, what will happen is that there will be accumulation of your phenylalanine, since the phenylalanine now is not broken down, no? So there is accumulation or build up of your phenylalanine, as I have mentioned, and this build up of your phenylalanine would eventually cause production of your toxic metabolites. Okay? Now, a defect here in the other end of the pathway, once your tyrosine, okay, due to the absence of the enzyme tyrosinase, once there is deficiency or absence of this enzyme, your tyrosine, on the other hand, will not be converted into melanin. And so, this will actually produce a person with no brown or black pigment, okay, giving a white appearance. No? However, that's actually abnormal. And then, if the tyrosine is not converted to melanin or any other metabolic products, what happens is that your tyrosine, again, since there is more phenylalanine now converted to tyrosine and the cycle or metabolism stops in here, what happens is that you have accumulation of a lot of your tyrosine, or there is build-up of your tyrosine, eventually leading to tyrosinemia. Okay? Now, or else, if tyrosinase is present, but another enzyme we call tyrosine hydroxylase is deficient or absent, your tyrosine now will not be converted into catecholamines. And so you will have deficiency of the different catecholamines. You will not have epinephrine, you will not have norepinephrine. Okay? And so there will be accumulation of your tyrosine, and this will eventually lead to build up of your tyrosine producing toxic metabolic products or tyrosinemia. Okay, so you need to memorize this figure and know what enzymes are affected and know what will happen if there is absence or deficiency in that enzyme. So we have several types of phenylketonuria. Okay, now why is this called PKU here? If we go back to this diagram, remember, in PKU, there is absence or defect or deficiency of your phenylalanine hydroxylase. And so, the defect on this enzyme would actually lead to accumulation or buildup of your phenylalanine. Now, a buildup of your phenylalanine would actually cause this phenylalanine now to be excreted more in the kidney or to be accumulated more in the blood. So, it has two pathways. It may go in the blood, and if this is accumulated in the blood now, you will actually have increased levels of your phenylalanine. But if your phenylalanine now goes into the urine, we call it now phenylketonuria, okay? Because you have a lot now of your phenylalanine now to be excreted out in the urine. So we have 3, 4, 5, and maternal types here. So type 1 or the classical type is characterized by a total deficiency of your phenylalanine hydroxylase. So this has something to do with the number, no, the amount of deficiency of that particular enzyme. Type 2 or the variant type is characterized by a partial deficiency of phenylalanine hydroxylase, whereas type 3 is characterized by a delay in the maturation of the enzyme. Type 4 is a deficiency of the protein, dihydrobioreductase deficiency protein. And type 5 is characterized by defective synthesis of dihydrobiopterin. And the maternal type is the genetic inborn error and wherein the mother has hyperphenylalaninemia. Okay? So, patients with classic PKU may appear normal in the first few months. However, they start to develop symptoms. So, presenting as seizures, tremors, or trembling, stunted growth, hyperactivity, eczema, and musty odor of their breath, skin, or urine. So this is a patient, no, with a PKU. So let's go now to the different tyrosine disorders. Okay? So remember, the source of your phenylalanine is your tyrosine. I have mentioned that. And your phenylalanine, if not converted to tyrosine, due to the defect phenylalanine hydroxylase defect, what happens is that there is accumulation in the blood. We term it as phenylalanemia. And if phenylalanine now goes to the urine, we term it as PKU or phenylketonuria. Now, if there's no defect in this stage of metabolism, all of the phenylalanine will be converted into tyrosine. And what happens now is that in tyrosine disorders, there is something wrong, a defect or a deficiency or absence of the different enzymes which will metabolize your tyrosine. Okay? Ice here. Now, usually your tyrosine is converted into several products. Okay? First, your tyrosine is converted into para-hydroxyphenylpyruvate. That is now made possible by the enzyme tyrosine transaminase. Now, absence of your tyrosine transaminase would actually lead now to increased levels of your tyrosine, accumulation of your tyrosine now, eventually leading to a specific type of tyrosinemia we call type 2, no, type 2 tyrosinemia. Or, if there is no defect in this stage and tyrosine is converted successfully into para-hydroxyphenylpyruvate and a defect is seen on the later part of the metabolic pattern. Now, your para-hydroxyphenylpyruvate is normally converted into homogentisate or homogentasic acid. And that is made possible by para-hydroxyphenylpyruvate dioxygenase. Defect, absence, or deficiency of dioxygenase of this enzyme would actually lead to another type of tyrosinemia we call type 3 tyrosinemia, causing now accumulation of your para-hydroxyphenylpyruvate, which is actually toxic to the body when not converted into homogentasic acid. Okay? Now, when there's no defect in this stage and your para-hydroxyphenylpyruvate is converted successfully into homogentasic acid, and if you go further into the metabolic process and this homogentasic acid should be converted normally into mallyl acetoacetate. If that is made now possible by the enzyme homogentisate oxidase. Any defect, absence, or deficiency of your homogentisate oxidase would actually lead to production or accumulation of your homogentisic acid or homogentisate. We call that now condition alkaptonuria. Okay? And furthermore, when there is successful conversion of homogentisic acid into mallyl acetoacetate, this mallyl acetoacetate is converted by mallyl acetoacetate isomerase into fumaryl acetoacetate. However, when these fumaryl acetoacetate are not converted into acetoacetate and fumarate due to a defect in the enzyme fumaryl acetoacetate, there is now accumulation of your fumaryl acetoacetate, which is actually a toxic byproduct, no? And that is now seen in condition called type 1 tyrosinemia, a defect in the enzyme 4-fumaryl acetoacetate. Okay? So let's discuss the common, no? The common tyrosine metabolism. Again, what enzyme? Okay, the homogentisate oxidase. Okay? So alkaptonuria is actually a rare genetic condition wherein when the patient's urine is exposed into the air, it turns into dark brownish black in color. Why? Remember, in your alkaptonuria, there is defect in the enzyme homogentisate oxidase. And when there is a defect in this enzyme, your homogentisic acid or homogentisate is not converted into mallyl acetoacetate. And so what happens is that there is build-up or toxic accumulation of your homogentisate or homogentisic acid. And your homogentisic acid has actually this chemical property that when exposed into air, it will actually be oxidized. And oxidation of your homogentisic acid will actually cause or lead to a production of a black compound. Okay, thus your urine now, when exposed to air in patients with alkaptonuria, will turn dark brownish black. So the enzyme that is defective here is the homogentisate oxygenase or dioxygenase. And as a result, homogentisic acid builds up in the skin and other body tissues. No? And when the urine is exposed into air, it turns brownish black. So look at the urine here. No? After the yellow urine, once the yellow urine is just sealed and not exposed to air, it will just remain yellow in color. However, if you open the seal up and expose this into air, it will actually cause oxidation of homogentisic acid and so producing a brownish black product. And if the excess of this homogentisic acid or homogentisate may actually accumulate also in the tissues, okay, giving the tissues a black lesions or black colored discoloration. Now another condition associated with tyrosine disorder is albinism. Okay, why albinism? So I mentioned this already. Remember, your tyrosine is actually converted to melanin by the enzyme tyrosinase. Now, your melanin normally is responsible for the dark coloration of the skin. And any defect in the enzyme tyrosinase would not actually cause conversion of your tyrosine into melanin, and so would lead to more and more tyrosine accumulating in the bloodstream or into the excreted out in the urine, and eventually leading to no or decreased production of your melanin. And since you have no melanin, which will actually impart coloration or pigment to the skin, we term that condition now as albinism. Okay? So this is characterized by a lower rate of melanin production, and what happens is that your melanocytes now actually do not produce the pigment melanin due to the what enzyme, what defect? Mutation, deficiency, decrease, or absence of the enzyme tyrosinase, which will convert tyrosine into melanin. Okay? And regardless of the amount of interference with melanin production, there's always associated visual problems in albinism. Okay? So these are the associated symptoms in patients with albinism, usually visual symptoms. So we have nystagmus, amblyopia, photophobia, hypoplasia, optic nerve misrouting, and astigmatism. And the test now to actually detect presence of the mutated gene, which would actually cause a deficiency or defect in the enzyme tyrosinase, is actually genetic testing. Okay. And another condition associated with tyrosine metabolic disorder is this what we call DRD, or your dopamine responsive dystonia. This is also known as Sigawa's disease or Sigawa's dystonia or hereditary progressive dystonia with diurnal fluctuation. So this disorder is characterized by limping. This basically starts as a defect in one of your limbs and resulting in a clubfoot and tiptoe walking. And then other characteristic symptoms of your DRD includes Parkinsonian features that are absent in the morning and worsening during the day with exertion. Okay? And this is usually treated with levodopa. So another conditions associated with your tyrosine include the tyrosinemia. Tyrosinemia happens when your tyrosine being not converted into the specific metabolite due to a specific deficiency of the enzyme. Okay, these tyrosine now will actually accumulate in the blood and so leading to a condition we call tyrosinemia. Okay, so this is usually inborn, in which the body could not effectively break down the amino acid tyrosine. And the symptoms include liver and kidney disturbances and intellectual disability. So we have two types, type 1 and type 2. So type 1 is also known as hepatorenal tyrosinemia, whereas your type 2 is also known as oculocutaneous, oculocutaneous tyrosinemia. So type 1 presents as in infancy as a failure to thrive and hepatomegaly. That's why it's called hepatorenal. There is involvement in the hepato, liver, and renal or your kidneys. The liver disease causes cirrhosis, hyperbilirubinemia, and elevated AFP, hypoglycemia, and coagulation abnormalities. Whereas your type 2 tyrosinemia basically affects the eyes. Okay? So, the symptoms include excessive tearing, abnormal sensitivity to light or photophobia, eye pain and redness, and painful skin lesions on the palms and soles. So this is treated by number one, a drug we call nitisinone and restriction of the tyrosine in the diet. So you avoid food containing tyrosine. And previously also, liver transplantation was the primary treatment option and is usually still used for patients in whom nitisinone fails. Okay, so another type of disorder is the catabolism of branch chain amino acid disorders. Okay, so what are again your branch chain amino acids? We have leucine, isoleucine, and valine. Okay, so what happens is that when there is an enzyme defective, what happens is that when there is an enzyme that will actually cause metabolism of these branch chain amino acids that become defective now, it will actually produce to a different or a series of disorders. Okay, so for example, the enzyme aminotransferases, which are found now in the heart, the brain, your adipose tissue, and your kidneys. This will convert your branch chain amino acids into keto acids. Now, another enzyme found in the liver, the heart, the brain, and any other type of tissue called the BCKA dehydrogenase complex will actually convert your branch chain amino acids into propionyl-CoA. Now, absence of this enzyme dehydrogenase complex would actually lead to the condition maple syrup urine disease. Now, if there's no defect in that enzyme, your propionyl-CoA is produced successfully from the metabolism of your keto acids. Now, further in the process, metabolic process, your propionyl-CoA should be converted into methyl-malonyl-CoA. Now, a defect in the enzyme responsible for that conversion, the enzyme is called propionyl-CoA carboxylase. This will lead to propionic acidemia. Now, your methyl-malonyl-CoA is subsequently converted into succinyl-CoA. Now, a defect in that enzyme, which will convert the methyl-malonyl-CoA into succinyl-CoA, the enzyme is called methyl-malonyl-CoA mutase, will lead to the disorder we called methylmalonic acidemia. So let's discuss the most common disorder associated with metabolic defect in the branched chain amino acid. No, the disorder is called maple syrup urine disease. Okay, so your maple syrup urine disease, the same with any other amino acid disorder, is an autosomal recessive metabolic disorder. Okay, now the term maple syrup or MSUD is actually derived from the urine which has a maple syrup odor. Okay, and infants with this disease seem to be well at birth, apparently well. However, when they get exposed to foods, when they ingest food now with branched chain amino acid, they become now quickly deteriorated. So at late onset, this usually presents with anorexia, weight loss, anemia, diarrhea, vomiting, dehydration, and lethargy. Okay, so usually the mainstay of treatment is to avoid foods containing branched chain amino acids, or you may infuse intravenous glucose as rapidly as possible. And usually for laboratory testing, we detect the amount of leucine, valine, isoleucine in the blood, via a mass spectrometry test. So another type of disorder, the last type, is a disorder in the amino acid transport. Okay, we term this condition now Hartnup disease. So what happens in Hartnup disease? So Hartnup disease is actually a defect in your tryptophan metabolism. Okay, now tryptophan is actually an important building block for protein and vitamin metabolism. Without tryptophan, your body could not produce niacin. Okay, so in Hartnup disease, what happens is that there is mutation in the gene that controls the absorption and reabsorption or metabolism of the amino acid tryptophan. And the symptoms associated with Hartnup disease is usually formation of a skin rash, called pellagra, and this may also appear in the face, the neck, and the hands, and the rash may progress to an eczema, and this is also associated with delusions, hallucinations, and etc. So in urinalysis, we test for presence of your tryptophan, okay? And we can also do a blood test, for detecting the presence of your vitamin B complex or niacin. So the mainstay of treatment here is to actually change the diet, or you let the patient, you do not let the patient eat any diet or food containing an amino acid tryptophan, because the patient would initially not able to metabolize the amino acid tryptophan. You also let the patient avoid sunlight and avoid sulfonamide drugs.